Welcome to the Best Practices for Regeneration webinar part 4. In the next few slides, we will discuss about the technologies that are being used in the screening strategies. I am Gianluca Spardella and I am a professor of medicinal chemistry at the University of Salerno, Italy. In the previous parts of this webinar, we already gave an introduction to heat generation, presented the screening approaches, and described the heat validation process. In this part, we will describe the most common technologies that are used in the screening strategies. It is important to note that practically any drug or probe discovery program will need to utilize multiple technologies and approaches to reach the eventual goal of a well-characterized probe or a therapeutic molecule. In this overview, for each technology we will illustrate the basic principles and provide a summary of the main advantages and disadvantages. In principle, we can consider the various assays and the technologies used for them from different points of view. For example, we can consider the target of the assays and roughly divide them into biochemical assays directed to isolated purified proteins and cell-based assays. Biochemical assays are aimed to measure the affinity of a ligand to a purified target protein or the effect of the ligand on a certain function of the protein, for instance the enzymatic activity in the case of enzymes. The readout of such methods could be based on measuring the intensity of fluorescence, luminescence or radioactivity signals, or on quantifying the level of specific modification using mass spectrometry. These methods are generally simple, high throughput, and may also give access to clear structure-activity relationships. Yet, their relevance to a specific disease could be limited. In fact, a ligand that resulted to be a good binder or a good enzymatic inhibitor may or may not enter the cell and may or may not affect the cellular function of interest. That's why these methods are complementary to cell-based assays and, if possible, both kinds of assays should be used to have a proper heat validation. As regarding cell-based assays, they are of course being done on cell cultures and are aimed at measuring a specific function of the target of interest or even downstream effects resulting from its modulation. But they can also measure the binding of a certain ligand to a target of interest in a cellular context. Again, the readout could be based on measures of the intensity of fluorescence, luminescence, or radioactivity signals, but also measures of variations of electric potentials are very common. Mass spectrometry could be used as well. The main advantages of cell-based assays include the higher biological relevance and also the possibility to go to several endpoints, for example, activity, toxicity, and so on. On the contrary, compared to biochemical assays, cell-based ones involve higher complexity and costs, usually a higher rate of undesired hits because of the complexity of the observed pathway or function, and the exclusion of low-permeable compounds. A few examples of biochemical and cell-based assays are provided in this slide. The various assays could be also divided in binding assays and functional assays. Both groups include assays done on isolated purified proteins and cell cultures. Binding assays detect the occurrence of the binding of a certain ligand to the target protein that could be isolated or in a cellular context depending on the method. Sometimes they also can measure the affinity. The readout could be based on the measure of heat exchange, absorption or release, protein stability, folding to unfolding transition, changes in molecular weight, and so on. Such methods may demonstrate target engagement and allow its quantification. Yet, as we said in the previous slide, the binding may not correlate with biological functions. As regarding functional assays, they measure the modulation of a certain function, for instance enzymatic activity, redox activity, or the downstream consequences of it. The readout is based on the measuring of the intensity of fluorescence, luminescence, or radioactivity signals, but also measures of variations of electric potentials are very common. Mass spectrometry could be used as well. These assays measure biologically relevant functions, but the observed readout may not be due to target engagement, since unexpected and undesired modes of action could also occur, and consequently false positives are not infrequent. An important group of functional assays deals with phenotypic assays. As exemplified in the pictures from left to right, they are aimed at measuring changes in structure and/or function of all cells, that's to say cell growth, morphology, viability, changes in structure and function of tissues, or changes in behavior or physiological parameters in animal models. We already dedicated our whole webinar to this topic, and you can have access to it using the link in this slide. And also, you may want to read the viewpoint paper we publish in Chem, as you can see in the reference in this slide. Now that we have seen how the assays could be classified, let's consider in details a few of the technologies employed for the various group of them. We will start from biochemical ones and consider the four most common technologies used in the classical screening strategies, that's to say fluorescence anisotropy, TR-FRET, alpha screen, and ELISA. We will consider the basic principles of them and the main advantages and disadvantages. Of course, this list should not be considered as exhaustive, but just as an example. Fluorescence anisotropy, also known as fluorescence polarization or with the acronym FP, can be used to measure the binding constants and kinetics of reactions that cause a change in the rotational time of the molecules. It is based on two principles: the fact that because of Brownian motion on the molecular level, each molecule has a kinetic energy and thus there are a continuous number of collisions between molecules causing rotation, and the principle that the light emitted by a fluorophore has unequal intensities along different axes of polarization. If the fluorophore is a small molecule, or if it is embedded into a small molecule, the rate at which it tumbles is significantly higher when it is free compared to when it's bound to a protein. Therefore, if a fluorescent ligand for the protein of interest is displaced from the protein binding site by a competing unlabeled ligand, the tumble rate significantly increases and can be observed. The advantages of FP include the fact that it's suitable for small ligands and that it can be used also in bioptic tissues or biological fluids of live animals. The cons are related to the limitation induced by the die lifetime and the size of the ligand. The larger the size, the smaller the observed effect, and the interference of autofluorescence events. For example, the compounds that are being screened could be autofluorescent and/or precipitate and scatter light, thus contributing to the background signal. Depending on the assay configuration, either case can lead to a false positive or false negative result. Time-resolved fluorescence energy transfer, or TR-FRET, involves two fluorophores, a donor and an acceptor. When such fluorophores are in close proximity, the excitation of the donor by an energy source, that could be flash lamp or laser, produces an energy transfer to the acceptor, which in turn emits light at its characteristic wavelength. The donor is typically a cryptate or lanthanide such as europium and terbium, possessing bright red shifted emission spectra and long fluorescence lifetimes, many orders of magnitude longer than background fluorescence or scattered light. Therefore, a delay in signal reading greatly reduces fluorescence interferences. The resulting assay provides an increase in flexibility, reliability, sensitivity, in addition to higher throughput and fewer false positive, false negative results if compared to other biomolecular screening assays, such as, for instance, fluorescence polarization. Although signal quenching may occur due to external interactions with the intramolecular excitation process, The main disadvantage is the need of lanthanide organic complexes capable to harvest and transfer light. There are many possible applications of this technology. Just to mention one of them, it is possible to run ligand-binding assays using a lanthanide-labeled antibody, the target protein, and a fluorescent ligand probe. The binding of an unlabeled ligand decreases the FRAC signal accordingly. It is also possible to measure enzymatic inhibition by using the enzyme substrate tagged with a fluorophore and a lanthanide-labeled antibody against enzymatic modification. That could be, for instance, methylation or phosphorylation. The higher the inhibition, the lower the signal. Alpha is the acronym deriving from Amplified Luminescence Proximity Homogeneous Assay, used to identify a bead-based technology for no-wash, non-radioactive proximity assays, technology that has become incredibly popular in screening strategies. It relies on the use of donor and acceptor beads that are coated with a layer of hydrogel which retains the dyes, minimizes non-specific binding and particle self-aggregation, and provides functional groups for bioconjugation. When a biological interaction between molecules brings the beads into proximity, a cascade of chemical reactions is initiated to produce a greatly amplified signal. The donor bead contains a photosensitizer that converts ambient oxygen to the singlet state upon laser excitation. The acceptor bead contains a thioxine derivative that reacts with the singlet oxygen to generate chemiluminescence. Singlet oxygen has a very short lifetime in aqueous solution. Therefore, in the absence of a specific biological interaction, the singlet-state oxygen molecules produced by the donor bead go undetected without the close proximity of the acceptor bead. On the contrary, if the biological interaction brings the two beads in close proximity, an alpha-screen fluorescent signal occurs. One advantage of alpha techniques is the fact that the beads are highly versatile and can be coated with various biomolecules, enabling detection of unique biological events. Virtually anything can be measured in a quick and easy way with alpha technology, including enzymes, receptor-ligand interaction, low-affinity interactions, second messenger levels, DNA, RNA, proteins, protein-protein interaction, peptides, sugars, and small molecules. Just to make an example, it is possible to have the substrate peptide of a certain enzyme linked to the surface of the donor bead and an antibody against enzymatic modification, that could be methylation or phosphorylation, for instance, linked to the acceptor bead. After incubation with the enzyme, the system is challenged with the potential inhibitor. The higher the inhibition, the lower the signal. Although alpha-LISA and alpha-screen assays are very valuable as primary screening tools, the alpha technology is somewhat prone to assay interferences, potentially arising from fluorescence quenchers, biotin or histidine tag mimetics, or light scatterers, which might lead to false positive or false negative hits. The fourth example of biochemical assay technology is enzyme-linked immunosorbent assay, very well known with the acronym ELISA and commonly used as the analytical biochemistry assay. In ELISA, a liquid sample containing the ligand is added onto a stationary solid phase with special binding properties and is followed by multiple liquid reagents that are sequentially added, incubated and washed, followed by some optical change, for instance color development by the product of an enzymatic reaction, in the final liquid in the well from which the quantity of the analyte is measured. The sensitivity of detection depends on amplification of the signal during the analytic reactions. Since enzyme reactions are very well-known amplification processes, the signal is generated by enzymes which are linked to the detection reagents in fixed proportions to allow accurate quantification, and thus the name enzyme-linked. The pros of ELISA include simplicity, easiness, high sensitivity, specificity and efficacy, as well as low-cost reagents. Yet, ELISA requires antibodies that could be labor-intensive and expensive to prepare, and also requiring refrigerated transport and storage. Another disadvantage is the high possibility of false positive and negative results. The first are mainly caused by hydrophobic binding of antibodies to plastic surfaces, but other causes could be non-specific reactions of the secondary antibody, ionic interactions between antibodies and compounds, interference with the enzymatic reactions, or poor attention in the washing step. On the other hand, false negatives could be caused by too low concentration of antigens, decreased immunoreactivity due to bad storage conditions, denaturation of enzymes conjugated to detection antibodies, denaturation of antigens, denaturation of antibodies by SDS, or inadequate protocol durations. It is important to point out that some of the technologies that are used for biochemical assays and that we just described in the previous slides can also be used for cell assays. In addition to them, we will now consider other four very common technologies used in cell-based assays: Nanobrea, Nanobrea, SATSA and ARTS, and Incell and Oncell Western assays. Again, we will consider the basic principles of them and the main advantages and disadvantages. Please remember that also in this case, the list should not be considered as exhaustive, but just as an example. Both Nanobrea and Nanobrea technologies are luminescence methods based on the small luciferase known as nanoluc, that is derived from a deep-sea shrimp and engineered to have high thermostability, glow-type luminescence, and a brightness that is more than 150-fold higher than firefly luciferase. Nanoluc also appears to have lower interference to inhibition by low molecular weight compounds. Although the overall signal strength for luminescence is much lower than fluorescence, the background signal is negligible for luminescence, often providing for a much greater signal-background ratio compared to fluorescence. Nanobrea utilizes a split protein system where 13 amino acids are removed from the carboxyl terminus of nanoluc to make a non-functional large bead, and activity can be reconstituted with 11 amino acid peptide of very low affinity called small bead that only bind to the large bead when in close proximity. Each portion of the split nanoluc is genetically appended to the protein of interest, and when the proteins interact, the small bead and large bead form a functional nanoluc. Due to the low affinity of the components, the tags do not interfere with physiological protein interactions, and as a result, the signal background ratio is high, reversible, and can be read in a kinetic manner. In addition, the small size of the tags is thought to have minimal impact on the tagged heterologous protein function and localization. This technology has been used to identify compounds that induce protein-protein interactions and compounds that disrupt protein-protein interactions, and has been recently demonstrated to measure post-translational modifications such as ubiquitination. Attention should be paid to the fact that high luminescence can saturate the detector, and therefore reduced gain or delayed reading is required. Nanobrea technology uses bioluminescent resonance energy transfer, or BRET, with the energy donor being nanoluc and the acceptor being a fluorescent ligand. It can be used to measure protein-protein interactions, but perhaps the most common use of nanobrea is to measure cellular engagement of protein targets with small molecules in an assay in which the ligand competes with a dye probe for the binding. The high-intensity light produced by nanoluc makes it an attractive donor, and the tail end of the bioluminescent emission spectra can be coupled to red-shifted acceptor dyes with almost no spectral overlap. In the case of cellular target engagement experiments, the target protein is tagged with nanoluc and a probe ligand labeled with red-shifted dye is used, giving rise to fluorescence. If the heat is able to engage the target, the fluorescence is proportionally reduced. This technology can also be used to screen ternary complex formation, such as with molecular glues and proteins. Just like nanobit, the method is highly sensitive and not affected by spectral overlap, but attention should be paid to the fact that high luminescence can saturate the detector and therefore reduced gain or delayed reading is required. One common question regarding the nanobit and nanobrea technologies is, since both technologies are great for measuring protein-protein interactions, which one is better? Which one do I choose for this application? Well, the answer is, it depends. In fact, the first thing to ask is whether your lab is familiar with resonance energy transfer assays and has the specific instrumentation available to detect this type of interaction. If this is the case, nanobrea could be the best choice, also because of the wide availability of ready-made assays. If not, nanobit assays only require a standard instrument capable of measuring luminescence. The cellular thermal shift assay, or SATSA, has emerged in recent years as a versatile target engagement and identification assay. It is based on the fact that proteins undergo denaturation upon a heat pulse and that ligand binding results in alterations of protein thermal stability. This often leads to an increase of the temperature needed for unfolding, thus causing the observed thermal shift. SATSA can be performed with intact cells, where following the heat pulse and subsequent lysis, the aggregated and denaturated proteins are removed by centrifugation, and target engagement is confirmed by the presence of an increased amount of protein for the compound-treated sample. The coupling of the technique to quantitative proteomics enables the profiling of thousands of proteins for the capacity to engage various types of small molecules. A key advantage is that SATSA doesn't require chemical modification of the compound of interest. It is readily adaptable to a microplate screening format for high-throughput target engagement studies, either through fusion to a reporter including enzyme complementation strategies, the use of alpha-LISA double antibody proximity assays, or an imaging readout. On the other hand, SATSA suffers from a few drawbacks, such as the tedious condition grouping process and insufficient target sensitivity, as well as the low throughput. A related technique is drug affinity responsive target stability, or DARTS, that is performed by treating aliquots of protein sample with the drug of interest and vehicle control, followed by limited digestion of the proteins in the cell lysate with proteases. And then the samples are separated by SDS page. Compared with the vehicle control, the protein bands that are protected from proteolysis by the small molecule binding could be determined. The protein sample could be purified protein or complex protein mixtures, such as total cell lysate, and the readout of SDS page could be Western blotting or mass spectrometry analysis depending on different purposes. Total cell lysate coupled with mass spectrometry analysis is usually applied in the experiment for drug target screening. The Incell Western assay is a quantitative immunofluorescence assay performed in microplates that combines the specificity of Western blotting with the replicability and throughput of ELISA. It is also known as cytoblot, cell-based ELISA, Incell ELISA, and FACE, fast-activated cell-based ELISA. It is based on standard immunofluorescent methods. The cells, cultured in microplates, are treated with the test compound, fixed, permeabilized, and stained with the primary antibodies. Then they are stained with the infrared dye secondary antibody conjugates, excited with infrared lasers, and the images are acquired with an imager and quantified. Oncell Western assays are cell-based assays that enables quantitative monitoring of cell surface protein expression. Like in Incell Western assays, they are performed in microplates, and the main difference between the two methods regards the permeabilization step. In fact, with the Incell Western assays, cell membranes are permeabilized after cells are treated and fixed, so antibodies can reach antigens inside the cell. However, the permeabilization process can compromise the detection of antigens present on the cell membrane. With Oncell Western assays, cells are not permeabilized because the protein targets are detected on the cell surface, not intracellularly. Instead, they are stained with antibodies against extracellular protein domains, so only cell surface antigens are detected. The cells in both assays can then be imaged either live or post-fixation. Both assays are fast and microplate-based, as we said, and they are characterized by high sensitivity and versatility. Yet, they both require a proper cell culture method allowing for an even distribution of cells across the well surface, and they are not useful where protein purification, concentration, or stoichiometric analysis are required. In fact, Incell Western and Oncell Western assays are restricted to the study of proteins present in high enough levels to be detectable from the cellular material in a single well, as it is not possible to concentrate low abundance proteins. Also, they do not allow determination of the stoichiometry of phosphorylation, such as can be obtained using urea glycerol page or via phosphate SDS page. Over the past 25 years, biophysical technologies have become key components of drug discovery platforms in many pharmaceutical companies and academic laboratories. There have been great improvements in the speed, sensitivity, and range of possible measurements, providing high-resolution mechanistic, kinetic, thermodynamic, and structural information on compound-target interactions. Biophysical technologies provide an important complement to data on biochemical and cellular activity and enable a more rational and rigorous problem-solving approach to the early phases of drug discovery. They differ in the physical principle that underlies the detection of binding, throughput, information content, sample requirements, sensitivity, and robustness of the resulting data. In the next few slides, we will describe the key biophysical methods, the information they can provide, and the main advantages and disadvantages. Two major microcalorimetry techniques are used for studying protein-ligand interactions. Isothermal titration calorimetry, or ITC, measures the generation or consumption of heat following titration of a ligand onto a protein, or the reverse. The data obtained include stoichiometry, KD, change in enthalpy, change in entropy, and the heat capacity change. Differential scanning calorimetry, or DSC, measures heat changes and corresponding thermodynamics resulting from the thermal unfolding of a protein. An advantage of these techniques is that experiments are performed in solution, but large amounts of protein are typically required. Mass spectrometry, or MS, is also used in different modes. Non-covalent MS, also called native MS, is usually carried out using electrospray ionization, generating data that can confirm binding, assess specificity through competition experiments, define stoichiometry, measure KD, and give an estimate of the enthalpy component of binding. Affinity selection MS, also called size exclusion chromatography MS, can be used to study protein-ligand interactions, but with decreased information content. Typically, a protein is incubated with a library of compounds and passed through a size exclusion chromatography column to isolate any compounds that are bound to the protein and therefore coelute with the protein itself. The protein complexes are then dissociated and the ligands are identified by liquid chromatography MS or electron spray ionization MS. In this case, confirmation of binding and KD values can be obtained. This method has successfully been used for the characterization of protein-protein interactions, such as those between epitope binding regions of therapeutic antibodies and their antigens. Furthermore, small molecule binding and alterations in protein dynamics can be investigated. The method works for soluble and membrane-bound proteins, and relies on an appropriate protease cleavage pattern to get accurate sequence coverage. The sample has to be desalted, and low-affinity binders are usually hard to detect. Knowledge about the 3D structures of biomolecules of pharmaceutical relevance and their drug complexes has proven to be of great value for an efficient and successful drug discovery effort. Structural biology provides a detailed view on the mode of binding of functionally active ligands and enables their structure-guided optimization. The most common technique used to this aim is X-ray crystallography, which can be used for proteins of any size. X-ray diffraction patterns of either protein-ligand co-crystals or apoprotein crystals soaked with a ligand are used to determine the structure of the complex at atomic resolution and derive the geometry and other details of protein-ligand interactions. Yet, the technique requires diffraction quality crystals that in turn require pure and stable proteins and the access to synchrotrons. Also, it is not a dynamic technique and doesn't take into account the movements of protein domains. High-resolution single-particle cryo-electron microscopy, cryo-EM, is a relatively new promising technology. Cryo-EM is used to study large micromolecular complexes up to 4,000 kilodalton, and some of these complexes reside in membranes, can adopt multiple conformational states, and or have been refractive to crystallization. Very recently, cryo-EM resolution has been narrowed to 1.25 angstrom for a few proteins, thus achieving the resolution needed to elucidate details of protein-ligand interactions and enabling structure-based design. Today, the minimal molecular size for high-resolution cryo-EM is about 200 kilodaltons, leaving a large set of relevant drug targets out of scope. This limitation can probably be reduced by future developments in methodology and instrumentation, or by studying molecular targets in the context of larger functional protein complexes. Microscale thermophoresis, or MST, is a relatively new methodology that monitors fluorescence in an infrared laser-heated spot. It is based on the detection of a temperature-induced change in fluorescence of a target as a function of the concentration of a non-fluorescent ligand. The observed change in fluorescence is based on two distinct effects: a temperature-related intensity change of the fluorescent probe, which can be affected by binding events, and thermophoresis, the directed movement of particles in a microscopic temperature gradient. Any change of the chemical microenvironment of the fluorescent probe, as well as changes in the hydration shell of biomolecules, result in a relative change of the fluorescence detected when a temperature gradient is applied, and can be used to determine binding affinities. MST allows measurement of interactions directly in solution without the need of immobilization to a surface, so it is an immobilization-free technology. On the other hand, it requires labeling of the target or strong intrinsic fluorescence. Nuclear magnetic resonance spectroscopy, or NMR, is another common technique that can be used to solve structures of protein-ligand complexes. But NMR also has a much wider application in probing ligand binding through two alternative experimental setups: ligand-observed NMR and protein-observed NMR. In the first, ligand-observed NMR, changes in the parameters, mostly chemical shift, relaxation rates, and diffusion rates of molecules or cocktails of molecules, are measured in the presence of a target protein. Typical experiments performed include relaxation editing, diffusion editing, and magnetization transfer experiments. The information that can be obtained can range from simply determining whether the ligand interacts with the protein or not, to binding specificity and information on the binding mode, such as epitope mapping, pharmacophore mapping, and mapping of ligand orientation. With some techniques, it is possible to estimate binding affinities. In protein-observed NMR, the chemical shift perturbations in the bidimensional NMR spectra of isotopically labeled proteins are monitored in the presence of ligands. Such measurements are limited to small proteins, practically those with a molecular mass lower than 40 kilodaltons. But when resonances have been assigned, the binding epitope and the dissociation constant can be inferred from the data. Both methods require relatively large amounts of a protein, which needs to be stable and of high purity. Surface plasmon resonance, or SPR, is a spectroscopic technique that monitors changes in refractive index at the interface of a liquid sample and a surface with an immobilized sensor molecule, typically the target protein. The signal is shifted as a result of analyte binding or induced conformational changes. When using microfluidic systems with continuous registration of the signal, it is possible to characterize the binding mechanism and determine the corresponding kinetic parameters, the association rate constant K-on, the dissociation rate constant K-off, and the affinity Kd. Thermodynamic parameters can also be estimated by performing analysis at multiple temperatures. An alternative configuration of SPR involves imaging of a microarray with immobilized small molecule sensors using a charge-coupled device camera, providing information on binding, yes or no, and binding specificity through on-array competition experiments. Both of these SPR techniques require immobilization of one of the binding partners, either the protein in the microfluidic system or a ligand library in the microarray technology. In the latter case, the protein is in the solution over the microarray, and thus, as the signal depends on the mass of the analyte, in this case the protein, the detection of binding in this setup is extremely sensitive. Thermal shift analysis, or TSA, also known as differential scanning fluorimetry, or DSF, measures the temperature at which a protein unfolds through binding of a fluorescent probe to the exposed hydrophobic surfaces. If a ligand stabilizes, or less frequently destabilizes, the fold upon binding, then there will be a change in the transition midpoint for thermal unfolding, positive or negative respectively. In some cases, it is also possible to determine Kd values. The major advantages of the method are its fast setup, its inexpensiveness, and the requirement of only small amounts of protein material. The disadvantages include the need of a fluorescent dye, the occurrence of artifacts, and the unsuitability for disordered or hydrophobic proteins. To conclude this part of the webinar, this slide summarizes the parameters that should be taken into account when selecting the right combination of technologies for specific screening approaches. Of course, information on the target are extremely important in terms of localization, overall knowledge, accessibility to the purified protein, but also previous literature reports on the same target or on targets in the same class. Also, the institutional know-how is extremely important. The accessibility to various technologies, the kind and the number of tool compounds that are available, and the number of compounds to be tested, as well as time and cost. So, the aim is to look for the best compromise between the throughput of the assay and the kind and the quality of the information you can get. You have to select complementary technologies in order to ensure the highest confidence in the hits and then adapt the assay cascade based on first results. Last but not least, it is important to have a good dialogue between chemists, biologists, and screening experts. In fact, the academic push to develop new chemical probes also continually pushes us into new areas of biology. This leads to the continual development of new screening technologies. You can be sure that the list of the technologies used in screening will continue to evolve to meet the needs of discovery.